we can start and see how it goes. So thanks for joining. The subject of this workshop is in QuPath for Fiji fans. So I'm Pete Bankhead. If you know me, you know that I'm involved in QuPath, well, created QuPath, but also a big fan of Fiji and ImageJ and used them for a long time before. And so, yeah, even though I'm very much in the world of QuPath at the moment, I still use ImageJ and Fiji all of the time. And so this workshop is really for people who, well, for people who like ImageJ and Fiji or maybe are familiar with it and are thinking about trying QuPath, or maybe already use both of them but want to know ways to sort of link together the worlds a little bit more. And so that's what we're going to cover in here. Feel free to ask questions in the chat and then we'll have plenty of time to discuss later on today. Okay. Oh, and I should also have said, there is a link there. So some of the material that we're going to talk about is already online there, and then we can add on more later. And as usual, the image.sc forum is a good place to discuss anything that comes up here today that we haven't time to discuss actually during the session. So this talk is going to be based upon QuPath 0.6, which, as you might have noticed, isn't really completely released yet. And so it's the RC3, so that basically means release candidate 3, so it's almost ready. It's just a few more things that we need to do or to finish off. But it's available, so you can download it, you can install it, and you can test it out. But I don't really recommend that you jump to do your analysis with this yet, because the final version of 0.6 should be released pretty soon. Currently, the stable version is 0.5, but there's more exciting, interesting stuff in 0.6. So that's what I want to talk about today, because it'll be along very soon. So this release candidate 3 was out just in time for the I2K meeting in Milan last week. Actually, it was released at the end of this workshop last week. So the workshop was used to test it, and because it survived okay, it was released at the end. So hopefully it will survive without any big crashes today as well. And so this is the link where you can download it, but you can also just go to the regular QuPath homepage and you can find a link to it there. So the main message for this, this talk or workshop today is, I think ImageJ and Fiji are great. So is Python as well, but that's really the subject for Alan's workshop, which is coming tomorrow. There are some tasks where I think that QuPath is probably a better fit. But I would say that if you can use both together, then it can give you kind of minor superpowers. I think it's quite satisfying to be able to recognize that there's something QuPath won't let you do, but you know how to do in ImageJ and Fiji, and if you know how to link them together in a sort of a quick and efficient way, then yeah, it's quite satisfying to be able to do much more than you could with either software on its own. And the idea goes a little bit back to Maslow's hammer, that it's tempting if the only tool that you know is, well, a hammer, or if your only tool that you know is ImageJ or Fiji or QuPath, then you might be tempted to approach everything just using that one tool. But by knowing different tools, then you can put it together and you can solve more problems in more creative ways. So within this workshop, one of the things that I would like to do at the very start is, assuming that you might be coming to this from the ImageJ Fiji background, not entirely convinced you should use QuPath at all, I want to say a little bit about, yeah, sort of some of what QuPath could potentially do for you and why you might want to consider using it. Then I want to describe one mental model that can help you make sense of QuPath. Because whenever you open it, you might see there's lots of different commands and lots of different options in there. But if you have this one model in your mind, then it can hopefully make sense of the entire software and be the key to becoming an expert in it. And then we're going to talk about sharing data among friends. So that's basically how we can share things between QuPath and ImageJ. And then I'm going to talk about the all new ImageJ script runner, which is the thing that was keeping me busy up until the very end, just before this presentation last week in Milan. So the first section then is why use QuPath at all? The easy answer could be because you have whole slide images, because that's, so the name QuPath, it comes sort of from quantitative pathology. It's not really limited to pathology, but if you work with pathology, you probably work with whole slide images and they're big and awkward and you don't really get support for them in ImageJ. But I hope to convince you that that's not the only reason to use a QuPath, and you don't have to be working in pathology in order to find it useful. So I created QuPath way back around 2014. It was only released in 2016, but around the time I was creating it, I was a postdoc. I really liked open source bioimage analysis software. I used ImageJ and Fiji every day, but I couldn't figure out how to make them work for digital pathology. And the trouble is that in digital pathology, you have these whole slide images. A single two-dimensional image could be easily 40, 50, 60 gigabytes in size. It's stored at multiple resolutions, so that's called an image pyramid, when you've got the same imaging data at different resolutions. And you need software which is able to go into these image pyramids and only pull out the tiles that are relevant for processing or visualization at that time, because if you try and read the entire 60 gigabytes in one go, then your computer probably just won't like it very much, because there's not really enough memory in most computers to be able to handle that. And even if you did have an enormous amount of memory in your computer, it wouldn't be terribly efficient to try and read all of it in one go. So I ended up creating QuPath in order to be able to handle these kind of images efficiently. And so it's been a couple of years since I gave a talk on this, on ImageJ and Fiji and QuPath. And back at that time in 2022, this is what would happen whenever I tried to open a whole slide image in Fiji. It would show you the different resolutions that you could choose from. And if you chose a low resolution, that was fine. But if you chose the full resolution like I did here, it might take a little bit of time and then run out of memory and you get an exception like this. Whereas if I take the same image and if I drag it onto QuPath, then it might take a few seconds to think about it, but then it can open up the image and it can sort of dynamically extract the pixels that it needs at any particular moment whenever you're visualizing the slide, and then also whenever you're processing it. And I guess some of you might have seen this before, but I'm assuming that a lot of you probably won't. The last thing that I'd like to do, just in the justification for why you might consider QuPath, is to show you a very quick video of 3 different types of analyses in 3 minutes. So all of these are focused on the whole slide images, but it gives you a kind of a flavor for what QuPath can do. So the first thing, so this is going very quickly, so don't worry about taking in everything here. But what I've done is QuPath will manage your images in a project. So I dragged an empty folder on top because I really like ImageJ's drag and drop. That created a project. Then I dragged 3 images on, that imported them in my project, and you can see them on the left. Currently, what I want to be able to do is to measure the size of the 4 different bits of tissue within this image. And so initially I used QuPath Polygon tool to draw. Now I'm using the brush tool. I'm not doing it very well because I'm in a rush trying to hit the 3 minutes. But then it gets a lot faster when I switch to the wands tool. So QuPath's wands tool is completely different from ImageJ's wand tool, but it's a very efficient way to annotate quickly. I created a results table, and what you should have seen there is that, unlike the ImageJ results table, you get little thumbnails of the things that you've annotated alongside the measurements visualized in QuPath, and we'll see later how that's possible. In this case, we want to measure, well, quantify in this like 30, 40 gigabytes whole slide image, the proportion of the tissue which is stained brown. We certainly don't want to have to annotate all of that. So I've drawn a few different examples of different regions in here, just a couple of different lines. One for what I think is a positive area. So it's originally brown, but it appears as red after it's been classified. And then partly as a negative area. And then QuPath is able to then generate a pixel classifier distinguishing between positive and negative. What you should see in the bottom left is it's giving me the percentage of the tissue that's positive, and it's actually eliminating the class that I told it to ignore. I can then adjust the resolution at which this runs at, because I said the original image is maybe 40 gigabytes in size. We don't necessarily want to have to process at that full resolution, so we were able to adjust that. And so again, QuPath is making it really sort of, well, quite fast and efficient to work with these huge images at the resolution that you need to. And then the last example is one where we're looking at cell detection, again in an image which is about 30 gigabytes in size. And so QuPath is a built-in cell detection algorithm. I ran it here. It detected just over 70,000 cells. It makes about 40 different measurements of them. You can visualize all of these measurements, these kind of dynamic measurement maps. You can even sort of share information about measurements amongst neighboring cells, so effectively smoothing out the measurements. And the reason why you might want to do that is because here we want to be able to distinguish between tumor and non-tumor cells, and then also between positive and negative. So positive is brown, negative is blue. And then the tumor cells tend to be a bit bigger, but they're not, size isn't on its own enough to tell you if it's a tumor cell or not. So what I've been able to do is use all of these measurements, interactively trained a random trees classifier, and then from this I can classify my 70,000 cells as being tumor, non-tumor, positive, negative, create these kind of density maps so I can find high density areas of positive cells or whatever kind of criteria that I want. And so, yeah, that's basically what we've seen in here. So I don't expect you to follow all of the details there, but hopefully it gives you a flavor of the kind of things that you can do very efficiently in QuPath. And just the whole interface and the way of working is quite different from ImageJ, even though it's actually very much inspired by ImageJ. And there was a few little bits in there where actually ImageJ was being used in the background. But QuPath is, quite distinct as well. Okay, so that hopefully gives you some sort of idea. And you might have noticed there's some little tricks in there which are similar to what you get in ImageJ, that if I, ImageJ, you often open an image by drag and drop onto the toolbar. You drag and drop onto the viewer in QuPath as well to open things or import them. And you might have seen it at some point, whenever you want to bring up a list of commands in ImageJ, that's Control L. In QuPath, it's the same shortcut. And so we try to make it easy to move between the two if you know one bit of software so you can go to the other. All right. So in a little bit, I'm going to then show you some of that live within QuPath. But firstly, I just want to give another few slides just on this sort of mental model that will help you understand everything about how QuPath is working and relate that to ImageJ so that you have the kind of the two different approaches to image analysis in your mind. So to begin, I want to say just a few words about what is the goal of image analysis in the first place. And I would say that one way to summarize it is that you want to reduce the number of numbers. So what I mean by that is if we take this K67 example, so this is the one where we have tens of thousands, if not hundreds of thousands of cells. We can see the nuclei and we want to distinguish are they tumor or non-tumor, and then we want to get the percentage of the tumor ones which are stained brown. So that's basically what we're trying to do with this image. But the image itself, it's about 28 billion pixels or so. So it's a lot of pixels and 28 billion numbers. So each pixel actually have three numbers, red, green and blue. So it's about 28 billion numbers is our raw data here. And that's what it looks like if we extract a region and visualize it at higher resolution. But what we actually want at the end is one summary measurement, which is the percentage of tumor cells which are K67 positive. So we want to go from 28 billion numbers down to one number. So that's a pretty dramatic act of compression or summarizing. And we can't typically do that in one step. So I could look at that and I could give you the number, that's 15%, but that doesn't really help you. There's no reason why you should trust me, because you want to have some kind of an intermediate representation that shows basically our working out for where that summary value came from. And so the goal of the analysis is to get to the summary value, but it has to do that with some steps along the way. And ideally we would be able to visualize those steps along the way to actually find out if we've done something sensible and not just picked a random number as the output. And so one approach that you might take to answer this kind of question within ImageJ is, well, firstly, I need to say you can't open the 28 gigabyte image in ImageJ, so I'm just working on a small crop at this point. But let's suppose we were to open that in ImageJ. What you might then do, or what I always do in ImageJ, is basically I duplicate an image because I don't know when I'm going to do the wrong thing and process an image and then want to go backwards. So often you would open your little image, your image like this, would duplicate it, and then you might apply some processing to it. So on the one side, I've extracted the red channel and applied a little bit of filtering, and on the other side, I've used a technique called color deconvolution in order to be able to digitally separate out the brown staining. And whenever you've done that, you might apply a threshold to one of your images and then run a command like analyze particles to be able to create regions of interest. And then you can even redirect the measurements so that here it's going to make measurements in the stain separated image, so that gets a measure of the brown staining intensity. I admit I'm not showing ImageJ at its best here because I just put together this algorithm very quickly, so it's not the greatest cell segmentation, I admit that. But the really nice thing here within ImageJ is you've got so much flexibility in the approaches that you can take, and then at the end you can generate lots of measurements and then start to, well, export those and then query it and turn it into your final results. In QuPath we can't do this. We can't even do the first step of it because we can't duplicate the image. So QuPath is written to deal with these huge images, and it can't assume that it's possible to hold all of the pixels in memory, nor can it assume that it's possible to copy the entire image, because if it did and the image is 40-50 gigabytes, you're going to be waiting a long time for the software to respond. So QuPath basically tries to work as much as possible without ever needing to do things like duplicate the image, and without giving you that kind of flexibility to separate channels and apply Gaussian filters and thresholds and so on. Some of those things are in there, but you're not expected to piece together those steps like that in the same way as you would do within ImageJ. And so what actually you do within QuPath is you start from your original image and then you try to come up with a way of representing the contents of that image in terms of objects. And you can think of objects as like regions of interest in ImageJ, but they're not actually the same, as we'll see in a moment. And if we can represent the contents of the image in this with these QuPath objects, then we can query that in order to be able to get our analysis results. We will see in a moment this in more detail, but basically an object within QuPath, it has a region of interest. It's not the same as a region of interest, but it has a region of interest, but it also has a list of measurements associated directly with that object. So if you think of an object as being like a nucleus, a cell nucleus, then it has a region of interest representing the bounds of that cell nucleus, but it also has a list of measurements like the mean staining intensity, the standard deviation, the area of the nucleus, the perimeter, the circularity, and so on. So all of those measurements can be added, sort of appended to the same object, and it can have a classification. So that could be tumor, non-tumor, positive, negative. It could be all sorts of different things. There are some extra properties that your object can have. It could be of a name and a color, but to be fair, you often don't need those. And it can also have relationship properties. So each object could have a parent or multiple child objects. So let's say you have this cell nucleus object. It could have a parent, which is like an area of tissue or a gland or a vessel, something like that. And it could have child objects, which could be like subcellular detections, like for subcellular structures or little dots or whatever. And so we have this way of representing the information with the image with the QuPath, and it's all based around objects, which are like regions of interest, but they're not exactly the same. But if you can build this object representation of the contents of your image, then this encodes the main information, and analysis is simply a matter of querying that. So here, for example, we go from my image with the nuclei, some of them blue, some of them brown, some of them tumor, some of them not. And then on the right, then I have the object representation of QuPath, where the colors correspond to the classifications of the different cells, and each cell is an object. And once we've done all of that, we have our objects, we have them classified. Then simply to get our percentage of positive tumor cells, we count up all of the cells classified as tumor, and then we count up all of the ones classified as positive, and then we use that to be able to calculate the percentage that's positive. And so all of the information is stored within our object representation, and the analysis at the end is simply a case of querying that. And so this is where it brings us to the one mental model to unlock QuPath. Is you can think of it, any analysis really in QuPath, in terms of four main steps. You have to generate the objects somehow, you need to measure them somehow, you need to classify them, and you need to query them. And so from our original image, here I've generated the objects, so each little red contour here. Actually there's two little contours, one for a cell nucleus, and then the one further out from that is for the estimate of the cell boundary. So together those form a single object within QuPath. But once we've generated those, then QuPath actually will measure them for you. So it will already, as I said during the video, it gave you like 44 different measurements just for free for each individual cell. So we get lots of the measurements for free. And then based upon those, you can use QuPath to be able to classify, in this case between tumor and non-tumor. And then further, we can classify even at multiple levels. So then I've further classified as positive or negative. So we have tumor, non-tumor, and positive or negative all applied to the same objects. And then once we have that, as I said, we can just we can count up, for example, the objects of particular classes. We can look up measurements of them, and then we can export those results. So we're basically querying our object representation. And the crucial thing really is that there's different ways to do each of these parts. So there's different ways to generate objects. I've shown you cell detection, but it's not the only way. There's pixel classification, there's superpixels, there's manual annotation, and as we will see, there's lots of tricks you can use in ImageJ to create objects as well. And knowing the ImageJ tricks gives you lots more options. There's different ways to create measurements, different types of measurements based upon shape, texture, intensity. And again, using ImageJ, you can calculate new measurements or different kinds of measurements and add them to your QuPath objects. There's different ways to classify as well. You can classify based upon a single feature. So for example, if we've got a measure of the brown staining intensity, we could say that if it's above, let's say 0.2, if it's above that, we say it's positive. If it's below that, it's negative. Or we could put in our 44 different measurements and we could train a machine learning classifier to make the decision and incorporate all of the different measurements within that. So there's different ways that we can classify based upon single measurement or based upon many measurements. And then there's different ways that we can query the results as well. So 2 questions when deciding whether to use QuPath is firstly, can you think of your analysis problem in terms of objects, as I've described? And if so, can you figure out how you would generate, measure, classify, and query those objects in QuPath? And if the answers to both those questions are yes, then QuPath's a good fit. Sometimes the answer is almost, and that's where, yeah, dipping into something like ImageJ can help you. And that's what we're going to focus on now. So generating objects is often the tricky bit, and if the built-in methods don't work very well, the built-in methods to QuPath don't work very well, the question is how can we figure out something else? And I think my last slide on this is, well, you would think QuPath is image analysis software. It gives you a list of methods to create objects, but it's not terribly good if you want to delve into the pixels and to do things like you can do really easily in ImageJ, like apply a Gaussian filter, threshold, and so on. QuPath isn't really for that. QuPath is really for generating the objects some other way with a built-in command and then querying them. And if you want to generate objects in some creative way that QuPath doesn't support, then you actually want to get your pixels into something like ImageJ because that will give you much more control. So QuPath isn't terribly good at working with pixels on its own, but ImageJ is, and so that's why putting them together can really help you. And that takes us on to this part on sharing the kind of information among friends. Okay, so at this point I will switch over and I will have a look then at showing live how some of this could work. So in case you haven't seen it before, this is what QuPath looks like when it's started up for the first time. You don't have to see this every time, you can turn this off, but it is quite useful to know that this will link you up to the documentation, to the forum for discussion, and also if you want to see the source code, you can see it because it's all open source. A few other options in here that you can explore if you want, but we will just get started. And so what I would like to do is I'm going to create a sample project. And to do that, I'll just create any empty folder on my computer. I will drag it on top of QuPath, and then QuPath will offer to turn that into a project for me, and you can see it under this project tab. Actually, a half hour after this workshop will finish, we will have QuPath for beginners, and so if you want any of this in more detail on this, then if you stick around, and then half an hour QuPath for beginners, we'll look at that in more detail. For now, we want to focus on the ImageJ side of it. So I am selecting then a few images and I'm going to drag them on top. Some of them are whole slide images, some of them you might recognize from ImageJ sample images. QuPath opens this import dialog. Let's just leave all the defaults and let it figure out what it wants to do. Whenever we do that, our images then are added to this on the left, on the project tab. And so, if you've been using ImageJ for a while, some of these should be pretty familiar to you. A couple of little things I would just like to show that might be quite nice features. This is QuPath's brightness and contrast command, and I've opened up this 5-channel image, and you can toggle channels on and off. So QuPath isn't limited to 8 or 9 channels, which I think is still the case with ImageJ and Fiji at the moment. You can really have any number of channels. You can't necessarily visualize them terribly informatively as a single composite image, but what you can do, and I'm going to use Control and L as the shortcut. Hang on, I'm going to have to, I should probably open up. I have a little command here because I'm so addicted to using the shortcuts, I'll probably forget to say when I'm doing it. So you can check this little window here in the bottom corner, and that will show whenever I use shortcuts. So Control and L, or Command and L because I'm on a Mac, will open up this command list, and I will look for the channel viewer. And this is a little QuPath tool which helps whenever you're working with images, lots of different channels, to be able to visualize them all simultaneously as you move the mouse around. And if you hold down Shift, then you can move the mouse without updating this display. If you have lots of channels, you can also like save the settings and then restore them later if you need to. And so some of those little tricks might be useful if you're working with this kind of images and you're currently doing it within ImageJ. You might find there's some nice ways to handle it within QuPath. But what we really want to get into is starting to link the two together. So I'm going to open this very familiar whole slide image. It's from the Open Slide Test dataset. It's under CC0, so everybody uses it for everything, and I'm afraid I'm going to use it as well here because it's quite nice as an example. And what we want to be able to do is take this image and then send a bit of it to ImageJ. So this is about 4 gigabytes in size, which is borderline possible for ImageJ, but probably not something that you want to try and open directly. It's about 46,000 by 33,000 pixels, so it is pretty big. What I want to be able to do then is to get part of this into ImageJ and then see what we can do with it. You might notice on the toolbar there is this little ImageJ icon, and that will allow us, well, let's say we're going to send a snapshot to ImageJ first. That's basically just a screenshot of what we currently had within the viewer. So we get all of the black bit around it, we get the little thumbnail image and so on, we get the scale bars and so on. So one thing that we can do is we can send a snapshot to ImageJ. But something that might be a bit better when it comes to analysis is that we can send an entire region to ImageJ, and we can choose at what size we want to send it. So we don't really want to send, if I choose the downsample value of 1, that'll basically try and send the full 4 gigabytes to ImageJ, and it's going to be quite big and it's not going to be terribly nice to work with. So I'm going to put in 10 here. And that's basically telling QuPath I want to send this image to ImageJ, but I want to scale down by a factor of 10. Let's see how that goes. Okay, so you can see here under the image tab, it was 46,000 pixels wide, and now it's 4,600 pixels wide. And so we're able to send that region to ImageJ. Why would we want to do that? Well, that then gives us the opportunity to start applying ImageJ commands. Oops, occasionally, yeah, it starts to complain and sticks the window in behind, so I should maybe stop using these shortcuts. So let's suppose that we want to then generate, just apply thresholds to this image, generate regions of interest, and we want to get these back into QuPath. So I've sent off my small region to ImageJ. Once I'm in ImageJ land, I can do all of the normal stuff. I can duplicate images, I can apply Gaussian filters, I can apply thresholds and so on. And I can do things like create selection. I should perhaps say that QuPath uses Java effects for the user interface and ImageJ uses Java AWT, and they don't always like each other very much, which is why I think the window's switching a bit which one's in focus. But what I can also then do from within ImageJ is run this command, which it's basically a custom plugin which QuPath will install. I should probably actually find it from the plugins. As I say, I'm quite addicted to the shortcuts, so that's my problem here. If I go to plugins send raw to QuPath, we have a region of interest here within ImageJ, and if I send it to QuPath, then what we should find is that the region that we generated within ImageJ is sent back to QuPath, but it's sent back at the full resolution. So even though we extracted this image and like 10 times smaller in each dimension, and we generated all of our regions here, QuPath, whenever it's receiving this image back, it's able to figure out exactly how much it needs to rescale, and then we can start to use ImageJ in order to be able to generate regions at a lower resolution and then send them back to QuPath. And so that's what we've done in here. If you're interested in how that actually works, so I pointed out that the region within, so the original image is 46,000 pixels wide, but we downsampled by a factor of 10, so it's 4,600. What QuPath has also done whenever it was sending the region off to ImageJ is it has set the properties. So it has also updated the pixel size information, which was previously very close to 0.5 microns per pixel. Once it sends it off at the downsampled resolution, it also adjusts these values according to the scale. And that's actually what QuPath is able to use in order to be able to figure out how much the image has been rescaled. And that's what allows it to send back the region at the right location. But ImageJ isn't just useful if you want to do things at a lower resolution. What if we were to extract a much smaller region here, but it's small enough that we actually could send it off? Maybe not the full resolution, let's downsample it a little bit. Actually, let's downsample it so it's 2 microns per pixel. We don't know exactly how much we need to scale it down, but we want it to be 2 microns per pixel by the time ImageJ gets it. So that's what I've done here. If I look at the properties, it is 2 microns per pixel. So QuPath has been able to rescale it. Again, I will just apply some simple processing. Oops, I forgot. I need to make this grayscale before I can do very much with it in terms of thresholding. Let's get a lower resolution region like that. And let's run the ImageJ command, create selection. And let's again go to our plugins, send the region to QuPath. And we should find that in addition to be able to fix all of the rescaling, it can also fix all the translation and get the region back to where it should be at the full resolution. And so effectively that's two things that we can do with ImageJ. We can send a low resolution version of the entire image, do whatever processing we want, and then send back what we get as QuPath objects. Or we can send a higher resolution region, cropped, send it off to ImageJ, and then still send it back and then have it updated if need be. Just showing again, it's not really doing anything particularly magical in there. It is still dependent upon the image properties. So I said it was 2 microns per pixel is what we asked for, and QuPath figured out how to resize it to make it exactly that by the time it got to ImageJ. But this origin value, which I don't know about you, but when I use ImageJ normally, I never really think about it. It's actually crucial here. This origin value is what is stored within the image and what is allowing QuPath to be able to figure out where it needs to put everything back at the end. And so that was used here. Okay. Something else that I should say though, whenever you launch ImageJ from QuPath, this is really just using a very small embedded version of ImageJ directly within QuPath. It only has the built-in plugins, but you might see that I have a few extra plugins that I've installed. The way in which that works is that you can set your local ImageJ directory, and whenever you do that, QuPath will make a guess as to where that might be on your computer. So if you have a separate version of ImageJ installed on your computer with your own plugins there, within QuPath you can set the directory typically to the plugins directory of ImageJ, and then whenever you restart QuPath, those plugins should be available to you within QuPath, or whenever you send the image to ImageJ. You can also set a Fiji installation. You can use the plugins directory of that. Not every plugin in Fiji is going to work because not everything is compatible with QuPath, but quite a lot of it should work. So that's how you can get some extra stuff in there. Another couple of things I would just like to show before moving on to the next bit of the presentation is, let's suppose I will open this mitosis image. So this is the regular ImageJ sample image. Now, if you go into ImageJ itself, and if you open this sample image, then let's suppose you do some analysis and you create a region of interest, you save it in the ROI manager, you're able to export regions of interest as this ROI set thing within ImageJ. And if I drag this on top, then ImageJ can open up the ROI manager, whoops, and hopefully, it's not so easy to see here. Okay, it is able to then import regions of interest which are stored separately from the image. QuPath also supports this, so if I drag this onto QuPath, then it will import from our ROI set the regions of interest. And so, yeah, QuPath is able to import ImageJ regions of interest. And you may want to do that, for example, to get one of these nice tables where you have the little thumbnail associated with your measurements and your histograms and stuff embedded within it. So you might find that even if you want to do most of your analysis within ImageJ, there are some benefits sometimes to be able to use QuPath to be able to visualize the results. And so that's why it's quite nice that we can work with this. Something else that you can do with ImageJ, oops, let's discard that, is that if you save a TIFF image, the TIFF image itself can also store regions of interest and overlays. And QuPath understands those as well. So if I drag this onto here with the QuPath project open, you might see there's a little import objects option in here. If I have that turned on, then these objects stored within the ImageJ TIFF are also brought into QuPath. If I import the same image and I turn off import objects, then they aren't. So this is with the objects from the ImageJ TIFF included, and this is with them excluded. And the last thing that I want to show in here is, let's suppose that I have a very small region. Oops, I should have said import objects. So this is a very small region. So I extracted this image that we saw before at a low resolution, sent it off to the ImageJ for processing. I saved it as a TIFF with a region of interest, and I can bring it back in. That's fine, but actually I would like to be able to resize these regions of interest and to get them at the full size. So how can I do that? Well, I can go in here, the original full size image. I can somehow open up ImageJ, it doesn't really matter how I do it. And then if I open up my small version, which has the region of interest embedded in it, I can do the send ROI to QuPath, and then QuPath is still able to figure out how much it needs to resize things, because all of that crucial information about the resizing is just stored in the image properties. That's preserved in an ImageJ TIFF along with the regions of interest, and so you are able to actually get information in there. So something that you might want to do is, if you have a Fiji plugin that isn't supported, even when you set the plugins directory in QuPath, you might be able to export your regions from QuPath, save them as ImageJ TIFFs, do all of your analysis in Fiji to generate your regions of interest, and then still find a way to get them back into QuPath afterwards if you need to. Okay. So I don't think there's very much to show on this next slide, except, well, yeah, that's what I've already showed really. It's all done through the Image Plus and the calibration. And yeah, some of the details of how this works, you can sort of tell from the QuPath documentation. How we have QuPath objects and ImageJ regions of interest, which are not quite the same things, but QuPath is able to, convert between them as best it can. The last thing then I want to show briefly, and then we can chat if we've got some time for that, is the all new ImageJ script runner, which didn't exist until very, very recently. And so I'm still learning how to use it myself, even though I wrote it, and I still figuring out exactly how to use it, but let's see. So if you've used QuPath and ImageJ before, you might be familiar with the macro runner, which it's been around from QuPath from the start. It is not very good. So let's go to QuPath extensions, ImageJ. So this is the old macro runner, and this is the script runner, and they look kind of similar, but I hope to convince you that the script runner is a lot better. So basically what this tries to do, it's a little bit like what we saw before, that you can extract a region within QuPath, you can send it to ImageJ, you can do some processing, you can send it back. So the script runner allows you to run macros to do this, and then to run it in batch, and then run it across multiple images or multiple regions in a much more efficient way, so you don't have to do everything manually. The macro runner did the same, but it was a bit horrible. Basic text editor, not very nice to use, whereas the script runner has some basic autocomplete if you can press control and space, and it has various examples, and it has nicer formatting and everything. So I will open an example called auto threshold. So if you used ImageJ macros before, this will look pretty familiar to you. And what I would like to do is to automatically detect these regions of tissue, but I want to do it using ImageJ's implementation of the triangle threshold method. So I'm just going to run it, and hopefully, yeah, already worked. Perhaps not brilliantly, but then let's switch from triangle to Otsu, and we try it again. And then, yeah, so we can get different results based upon that. I can then start to then make adjustments to the macro. So let's say I want to run with a bit of filter in here. So this is all basically just exactly what you would get with a normal ImageJ macro, but then running within QuPath. So what we need to talk a little bit about then is how this actually works with these options down here. So these basically control what exactly is going to be sent off to ImageJ. Similar to when we sent a region to ImageJ, there's different ways that can define the size that it should be. We can say a fixed amount that we want to scale down by. We can say the target pixel size that we want to have to resize to an ImageJ. But the easiest way to make sure we don't send a region that's too big is to set the largest dimensions. So basically QuPath is going to resize the region that's being sent to ImageJ to make sure that it's as big as possible without the width or height being above this value. But you can choose one of the other criteria if that's what you want. You can also control which channels are being sent. So at the minute I sent the red, green and blue values, and if I press test run here, you can see that ImageJ briefly, it got the red, green and blue values. But what I could do is I could say that I only wanted to send one channel, and with test run you might have briefly seen that it was grayscale. And so this gives you a chance to control what is being sent to ImageJ. You can also optionally send the region of interest, which is currently active in QuPath, to ImageJ. So I'm going to have to switch to send the selected object. I'll send it to ImageJ, and oops, that was not very obvious exactly what happened there. So that probably didn't help us an awful lot. But yeah, wasn't the best example to show. But you should have hopefully have seen that very briefly as that appeared, we ended up with the region that was selected. Actually, I may be able to show that in a slightly more convincing terms if I draw a region like this, and if I run it from scratch, you can see that it has been able to send off the ellipse to ImageJ, and then the region that you get back is cropped to that. But then you can send like very exotic, awkward shapes and get the same sort of results. So I'll select all of these, and then I will run the script. And then hopefully what you'll get back is the regions detected and cropped accordingly. And so I recommend that you spend a bit of time, if you're using this, sort of play around with this, because there's various things that you can do. You can send back the currently active region of interest within ImageJ, but you can also use something like Analyze Particles to create an overlay in ImageJ where you've got lots of different individual regions, and then send them all back as QuPath regions of interest. So that's what this overlay thing is for. So you can bring back overlay information to QuPath as well. And then you probably want to delete anything that's currently within the region that you're sending, otherwise you can end up with duplicates. But all of these things are there as options, and there's tooltip text if you put your mouse over it, or if you click on this little help button up in the corner, then throughout QuPath, anytime there's extra help text, that command up here can help you find it. You just put your cursor over it and then you can read it more quickly than having to wait until the tooltip appears. Okay, so that is the basic idea of how we can start to use the ImageJ macro runner. What I guess I should point out very quickly then, and then we'll end so we have time for questions, is there are some extra tricks hidden in here. So if you're using ImageJ and if you set a threshold, you often see there's this little dark background checkbox. Whenever you work with an image in QuPath, you normally have to tell it, is it bright field or fluorescence? That's the same kind of information. So a black background or dark background often means you have a fluorescence image. So QuPath actually tries to put that information into the properties of the image, which means that you can query it in your macro. And so that's why if I switch over to a completely different image, so here we now have a fluorescence image, but hopefully what we should find, I'm going to choose a more informative channel to threshold, is that exactly the same automated thresholding macro works. We don't need to adapt for dark or light background because actually that has already happened within the macro, because QuPath has passed that information that it already knew in, and the macro is automatically adapted to the sort of background that it has. And so hopefully we're going to add in more of these sample scripts that show more examples of that in action. And then you can see how some of these tricks work. Here's just another little one, which I did not try in advance of this, so it's slightly risky to try it now. So fairly terrible in terms of results, but the point here is that we could run a little like maximum or minimum detection command. We could run that in here in the same sort of way. So there's different macros in here to do quite different things, including even classifying objects. And also, as a little bonus, you can even run groovy scripts within this same macro runner. And so I recommend playing around with that and seeing if it does anything sensible, depending on what you might want to do. Okay, so here I've sent back multiple regions on an overlay, and so that kind of shows you that that is possible. It's very tempting for me to just to play around with this for a while, but I should probably stop in case you have any more meaningful questions, and I'll stop playing with it myself. So, try and look at the chat if I can figure out exactly how to work Teams again, or work. That was a bad mistake. To work Zoom. Okay. Okay. So if I just start from the beginning in the chat, and feel free to add more questions. The first question is, can you run a QuPath model in an ImageJ macro? I don't know, do you want to elaborate on what you mean by a QuPath model in this sense? So I'm not sure what the answer to that is. The model would classify. That's an interesting question. I've never tried. So what I showed you here briefly is how you can run like the ImageJ macro language and stuff within QuPath. You can also run scripts directly within QuPath which operate using ImageJ. And so my guess is if you want to run like a QuPath classifier, probably the macro runner is not the way to do it, because we don't have an easy way to do it. But you could potentially run a QuPath script which can run a QuPath classifier and run an ImageJ macro at the same sort of time. So I'm not sure if that's exactly the workflow that you want. It sounds like something that should be possible, but not necessarily in the sort of easy way of you run a QuPath classifier from an ImageJ macro. Okay, The next question is, in QuPath, how are the cell boundaries determined without membrane staining? Excellent question, because it reminds me of something that I should have said. Well, the first answer is not very well, just by distance-based expansion. So with QuPath's built-in cell detection, it will try and detect the nuclei, and then there's a parameter in the cell detection about expansion, and it will expand out all of the nuclei until they reach that limit, like 5 microns, or until they hit a neighboring cell. And so it's purely a distance-based expansion, which isn't, it's okay for bright field. It's useful for a classifier, even if it's not very accurate, because it can give you informative measurements like nucleus to cell area ratio, which helps you distinguish tumor cells from non-tumor cells. And so it's a useful measurement, even if it's not a terribly accurate measurement in terms of it doesn't actually give you the proper membrane. But tomorrow, Thibaut from the group, Thibaut Goldsborough, he is going to speak about InstanceEgg, which is a entirely new and different deep learning based method to cell segmentation, which runs through Python, but also is a QuPath plugin for it, or QuPath extension for it. And that is able in fluorescence images to use basically any combination of channels and try and make a much more informed estimate of the cell boundary. You can also use it in bright field images, but we don't have such good training data sets in order to get it to do it. But in principle, it's able to use so much more information within the method and within the image. And the method is called InstanceEgg, and Thibaut will speak about it tomorrow as part of these workshops. So I recommend joining for that. And we might also cover it briefly in the beginners one that's coming up later this afternoon if we can. I see Alan has already mentioned that. Alan from the team. Okay, does the script runner work with Groovy scripts too? Yes, it does. Because it was happening very late, there is no actual option to choose the language that it runs. So what it will do is that if you're running a Groovy script, you normally have to use this command like IJ get image to get the current image that ImageJ has. And if it sees that, it'll recognize that it's Groovy. And so it's not, it should automatically sense when you've got a Groovy script, but it's not terribly smart. But yes, it can do the ImageJ micro runner or Groovy scripts, both. I don't know if that's, I should really listen to Robert as to how that's pronounced. Is it CLIJ? Is it supported by the ImageJ script runner? No, at least it's not something that we've tried. I don't know. I know there was QuPath at a time. So basically this is a way that Robert has written about using like GPU accelerated stuff within ImageJ and Fiji. At one time he had a QuPath, like a way of linking it up with QuPath, but I'm not sure of the status of that now, but it's not something that we've explicitly tried to do. Because QuPath is better at 2D than 3D stuff, I think probably there's less to be gained from that, because when you're working with 2D things, it's not really the computational stuff that's the bottleneck. It's very often like reading the image regions. It's different with deep learning because then you've got so much computation that the GPU helps. So yeah, I suppose to summarize, I'm not sure if it's possible, and I'm not sure if it would be massively useful because QuPath isn't as good at 3D, and I think that the biggest impact you see with that work is in 3D. But it might be a Robert question, not a me question. When should we be using the ImageJ script runner versus the original micro runner? I would hope that you will never have to use the original macro runner. At the minute it's a release candidate, so if you have a context where you would rather have the original macro runner for some reason, then you can let us know, but I'm hoping it can completely replace it because it can already do almost everything that the macro runner could do, and a lot that it can't do. And if the few things that it can't do that the original micro runner could do that are important to you, then let me know and we can try and find a way around it. But the script runner, one of the things that it will do, the new one, is it can add your script to the workflow, and then you can run it in batch in a Groovy script and so on. So I think it's much better. So hopefully you will find that you never need to use the old macro runner, but If that's not the case, we can discuss it in the forum. Okay, when would you use ImageJ from QuPath? Thresholding is already easy in QuPath, so I'm glad. Thresholding is easy in QuPath if you have a fixed threshold. If you want to have an automated threshold based upon the image histogram, that's where ImageJ is better, because we don't really have a nice way to do that in QuPath at the minute. So like the triangle method, Otsu's method, they can all sort of automatically adapt. And so you might find that they are more useful than QuPath's built-in fixed threshold sometimes. You also might find that there's a more complex macro in ImageJ beyond thresholding that you find easier to write in ImageJ, but yeah, it's not something, yeah, it's probably going to be most useful to you if you're very comfortable using ImageJ and you just can't find QuPath commands to replicate it, or maybe there aren't QuPath commands to replicate it, like with the automated thresholds. The last question I see here is, does it work with side Java parameters, or is there any plan to? The answer currently is no, and I'm not sure that Curtis would be happy with that. Yeah, it's something that we would look into and see whether it's something that we can do. But yeah, it's not possible at the moment to be able to work with side Java parameters. So that's very much a Fiji thing. One of the things that Curtis announced in Milan last week is about Fiji working with Java 21, and QuPath uses Java 21. So the fact that they use the same Java version will potentially make it possible to link them together in ways that previously wasn't very easy to do. But it's not something that we've explored because it's all Fiji moving to Java or supporting Java 21, and this existing of all just happened in the last few days. So, okay. Are there any other questions? Okay, is the macro runner described the same thing as using the run button in the editor? No. Okay. So QuPath is a script editor, and then you can just generally, you can write a script entirely in QuPath and then you can press run. And basically that'll give you access to everything in QuPath's way, like QuPath's way of extracting pixels and so on. And so using that, I have some examples on the GitHub repo where it shows how you can create a Groovy script within QuPath, and it will extract pixels, it will convert them into an ImageJ friendly format, and then it can apply some processing, and then it can bring it all back to QuPath. So that would happen through QuPath's normal script editor. The thing that's different about the macro runner is that the macro, or the new script runner, is that basically in the background, QuPath will do a lot of the hard work for you in order to be able to extract the image, convert it into an ImageJ format, even put it in the right thread if you want to run it in parallel processing. Convert all your regions of interest, and all of that happens automatically without you having to think about it. So you only have to write an ImageJ script or ImageJ macro that basically asks what's the current image, and that will just be a little region that QuPath has given it. Do your processing on that, and then QuPath script runner will figure out what to do with the results. So the amount of code that you write is a lot less using the new script runner, and you only focus on the processing part. But the cost of that is you don't have quite as much control as if you write a script end-to-end entirely in QuPath. You get more control, but you have a much more complicated script usually. So yeah, I don't know if that, hopefully it answers your question. The kind of thing that you benefit from the new script runner is, let's suppose that you wanted to even detect something, do a subcellular detection in 100,000 cells. You could write a tiny little bit of code that runs for one cell, and then run it through the new script runner, and it can parallelize it, it can send all the regions of interest and do all of the stuff, the tricky, annoying stuff in the background, and you would only have to write the code for one cell, and then the script runner would let you do it. Whereas with our regular QuPath script editor, you have to manage all the cells, request them and everything, and do lots more boilerplate stuff, which is hard to write. Okay, thank you for the thank you. And can you call an ImageJ macro in script runner? I don't know. if you can call an external ImageJ macro. Haven't tried that. The answer might be no. So yeah, this is really new and I've barely used it myself, except to demo it. So it's very possible that there are important things that we should add to it. So yeah, you can use the image.sc forum, be able to chat about that. Say a macro in the macros folder, Fiji, don't know the answer. So give it a go and let me know in the forum how it works. And yeah, try and figure it out if it's not possible at the moment. Okay, so I'm gonna end this, I guess, for now, and in half an hour Fiona will be back and I'll be back as well for the QuPath for Beginners workshop, so hope to perhaps see some more of you there. All right, thanks for joining.